Hello, everyone, and welcome to another episode of Dr. Gilbert Hosts. Today, our broadcast is entitled, Taking Research from the Labs to Our Lives. We're going to be discussing two research projects funded by the American Parkinson's Disease Association. And we're going to be focused on how these research projects may have direct implications on the lives of people currently living with Parkinson's disease. And in order to do this, we have 5 special guests who are going to help us in our discussion. So I'm very excited to introduce those 5 guests. Our first is Dr. Nicole Bentley. She is Assistant Professor of Neurosurgery at the University of Alabama School of Medicine at Birmingham. We have Dr. Daniel Weintraub, who is Professor of Psychiatry at the University of Pennsylvania School of Medicine. We have Dr. Mohammad Shahnawaz, Assistant Professor of Neurology at the University of Texas Health Sciences Center at Houston. Dr. David Standaert, the John N. Whitaker Professor and Chair of the Department of Neurology at the University of Alabama School of Medicine at Birmingham. And Dr. Kevin McFarthing, a biochemist and person with Parkinson's. I want to welcome everyone to our broadcast. So here is our lineup today. Dr. Nicole Bentley and Dr. Daniel Weintraub are going to explore a really fascinating possibility, the possibility of using deep brain stimulation techniques to improve cognition in Parkinson's disease. After that, Dr. Mohammad Shahnawaz and Dr. David Standaert are going to explore a potential for a blood test to diagnose Parkinson's disease. And after those 2 presentations, Dr. Kevin McFarthing, who is a biochemist living with Parkinson's disease, will discuss the vital importance of research for those living with the disease. So let us begin. Dr. Bentley, can you tell us about your exciting APDA-funded research?
Yes, thank you very much. And thanks to all of the people viewing. I'm excited to talk to you today. So my research grew out of the observation in patients that I treat, primarily with deep brain stimulation, that the therapy of deep brain stimulation is very helpful for their motor symptoms of Parkinson's disease that are so prevalent and disabling. However, a lot of my patients, though their motor symptoms are very well treated with DBS, they continue to have a lot of difficulty with cognitive function. And cognitive deficits in Parkinson's disease outpace what is seen in the general population. And for this aspect of PD, there are very few treatment options, and DBS itself is often not helpful or can even make cognition worse. And so in deep brain stimulation, what I do primarily is to implant a device. There is basically a wire that goes into the brain to target structures deep inside. And by delivering electrical stimulation, it can improve motor symptoms. But in order to study and understand cognitive function, we have to look at the frontal lobes primarily, which is shown here in the red box. And one aspect in particular that I'm interested in is called response inhibition. It's a type of cognition that involves the ability to stop actions that are ongoing. So the example that I give is if you were trying to cross a busy intersection in a crosswalk and you were getting ready to start that action, but then looked to the left and saw a car coming, the ability to stop that action is called response inhibition. And studies have shown that patients with Parkinson's disease have a lot of difficulty with this particular aspect of cognition. And so my research is aimed primarily at two different aims. The first is what happens in this prefrontal region of the brain during a cognitive task of response inhibition, with the rationale being that if we can measure these changes in brain activity, we may be able to use that information to develop treatment. And the second aim is, can we deliver a type of stimulation that is helpful for cognitive impairment, since we know that stimulation delivered for motor impairment isn't particularly helpful. So we wanted to find out if other patterns could potentially improve cognition. So to explore the first question, of what happens to prefrontal activity during a cognitive task. We first, during surgical cases, place the deep brain stimulator electrode as normal, and also place a separate electrode that records brain activity. And this is placed over that frontal area that I showed. This is temporary. It comes out at the end, and basically just gives us the ability to record from two different areas during a task. So here you're seeing an X-ray from the side of the head with the blue box showing the deep brain stimulator electrode at the target, and the blue oval showing the other recording electrode. And then on the right side of the screen is an example of the task, which for this particular area of cognition, we use the go, no-go task, where you're instructed to press a button for any letter other than X. And so when you see an X, you have to withhold that response. And the task is set up so that you have the urge, so to speak, to press the button. That tests response inhibition. And so what we see when we look at those recordings and analyze that data is that when patients who are performing this task, when they see that go response, which is any letter other than X, we see in the black dotted line a recording of muscle activity. So you can see sort of the mountain peak there. That represents the button press itself and the muscle activity as they're pressing that button. And we see some increase in a particular type of brain activity in what's called the theta range. And that's shown by the increased yellow that you see right below that peak. When they're withholding that response, you can see in that bottom panel, the muscle activity doesn't have the same peak. So they're not pressing the button. They're sort of maybe twitching a little bit up and down, trying to decide. But at the same time, we see a very stark increase in that same theta region of activity. And on the right side, that's sort of highlighted here through statistical tests, which show that that is indeed a statistically significant increase in power. And so what this suggests to us is that activity in this range of function may have something to do with the ability to withhold a response. So in order to explore the second question of can different patterns of stimulation be helpful, we are trialing something called intermittent theta burst stimulation. And this is still in the early stages, but essentially what this does is, unlike the typical stimulation used for Parkinson's disease, theta burst stimulation delivers bursts of electrical stimulation at that same frequency, which I was just talking about. So in the middle panel, sort of a blue panel there, you can see the increase again as the yellow in that range. And in our first several people that have tried this, we do see an improvement in their response and their behavior during that stimulation. And that's shown on the right side of the screen with the lines going downward, which shows an improvement in response. So this is early stages. There's lots more to do. We are continuing to enroll participants for this study and specifically want to look at cognitive impairment versus those without, and to try this stimulation in a more chronic setting outside of the operating room. So those are some next steps for us. And I would like to thank all of our team who are helping with this study, and the APDA as well, the University of Alabama. And with that, I will turn it back over to Rebecca.
Thank you very much for that fascinating exploration of your research. I now invite Dr. Daniel Weintraub, who is an expert on cognition and Parkinson's, to comment on this research and tell us, can we expect deep brain stimulation to be used in the future to help cognition for Parkinson's?
Very interesting description of your research, which is not hard to describe in a few minutes and to convey. It's very complex material. So I just wanted to take a little bit of a broader view and say that I think modulation or stimulation therapies really aim to target impaired connections between brain nerve cells, both at the local and what we call the network level, to have wide brain effects. And they have persistent effects on particular brain chemicals in cells that are associated with cognition, and including the growth of nervous tissue in the brain. So with that kind of limited background, different stimulation therapies are used and being tested for cognitive impairment in various neurodegenerative diseases, whether it's Alzheimer's disease or Parkinson's disease. The most commonly used one to date has been external direct transcranial current stimulation, or DTCS as it's called, which is an electrical stimulation to the frontal part of the brain outside of the brain that Dr. Bentley was describing. And sometimes that's paired with what's called cognitive training, where the patient will be participating in a cognitive task at the same time. There's also external intermittent theta burst stimulation. Dr. Bentley was describing internal inside the brain, but external intermittent theta burst stimulation, or ITBS as it's called, which is really a modification of a more commonly used procedure in psychiatry called repetitive transcranial magnetic stimulation, or RTMS. And this also to the front part of the brain has shown some preliminary benefit in Alzheimer's disease and Parkinson's disease cognition research. And I think the main difference, my understanding of how ITBS, intermittent theta burst stimulation, differs from repetitive transcranial magnetic stimulation is that it uses this lower frequency that Dr. Bentley was describing, and it also has a shorter total duration of treatment exposure required. So two differences there. When you look at the bigger picture of DBS, deep brain stimulation, and cognition in Parkinson's disease, I'd say overall, the literature seems to suggest that these procedures are associated with slightly worse cognitive outcomes when used clinically for the treatment of motor symptoms. But if you look at cognitive performance 6 to 12 months later in particular, you might see a slight decline on average in the patients that undergo deep brain stimulation surgery. However, when patients are younger and have better cognitive performance at baseline, the impact of the DBS on cognition seems to be more neutral overall. There's also been a very recent study showing that it may be possible to change the stimulation site within the subthalamic nucleus, for instance, where the stimulator is, or the electrical stimulation is placed, to decrease cognitive decline after surgery. It's also possible that lower frequencies overall may be associated with better outcomes. So in terms of moving forward, and I'm sure Dr. Bentley and others involved in research in this area are thinking of these research questions, I would want to know, are there other cognitive abilities besides those linked with the front part of the brain that might be targeted, for instance, memory, with this kind of a procedure? I would want to know, beyond changing the frequency of the stimulation, which is really the focus here, what about the location within the subthalamic nucleus or the globus pallidus interna, which is the other major site of deep brain stimulation surgery? What's the potential impact of these very low and altered frequencies on motor function, which is the primary reason that Parkinson's patients get deep brain stimulation surgery to begin with? And then finally, another question would be, is this envisioned as an actual treatment for cognitive impairment in patients with Parkinson's disease? That is a reason to get the surgery, or is it really more conceptualized at this point as a way to minimize the cognitive decline that can occur in some patients after DBS surgery? In summary, though, I'd say that the area of stimulation therapies, either external or internal to the brain, as a treatment for cognitive impairment and neurodegenerative diseases is an exciting area of research and one worth exploring further.
Fantastic. Thank you so much for those comments. Obviously, a very exciting topic to explore, but a lot of questions and not yet ready for prime time. So thank you both, Dr. Bentley and Dr. Weintraub. We're going to go to our second APDA-funded project. Dr. Shahnawaz, if you join us on screen, can you tell us about your exciting APDA-funded research?
Hi, everyone. Thank you very much for joining us. I'm really happy to share my research. I'm going to talk about a blood test for Parkinson's disease that we are working on. The question is why a blood test is important for Parkinson's disease. One of the main problems for developing a disease-modifying therapy for Parkinson's disease is the lack of early diagnosis. It is too late by the time disease is diagnosed. As you know, that currently there is no test available. A blood test offers several benefits, such as it is non-invasive, inexpensive, and can be used as a routine test. Now the question is, what we are detecting to diagnose Parkinson's disease? One of the key changes that happens in the brain of Parkinson's patient is the accumulation of alpha-synuclein proteins in the neurons, also called as Lewy body. Over time, these neurons die and disappear, which results in the onset of clinical symptoms. The formation of these clumps starts years or even a decade before the onset of clinical symptoms, in which a normal alpha-synuclein, which is a synaptic protein, forms small clumps, also called as oligomers, and then forms large clumps, also called as fibrils. These oligomers are soluble, highly toxic, and can be found in biological fluids, such as cerebrospinal fluid, blood, saliva, and urine. However, the quantity of these oligomers are too low in biological fluids. Because of that, the oligomers cannot be detected easily. Now the question is, how we can detect these oligomers? To address, we have adapted protein misfolded cyclic amplification assay to detect these tiny oligomers present in biological fluids. Here, our main aim is to increase the number of alpha-synuclein fibers using protein misfolded cyclic amplification and detect these fibers easily. The PMCA assay is a cyclic process which combines two steps, as you can see here, incubation and fragmentation. In the first step, the normal protein, which is alpha-synuclein, is incubated with a biological samples such as cerebrospinal fluid, blood, saliva, and urine containing oligomers. That allows the formation of long fibers. In the next step, as you can see here, these fibrils are broken into small pieces. That results in the formation of large number of short fibers, which act as nucleus. After several cycles, a large number of fibrils are formed, which can be easily detected using fluorescent dye. As you can see, a typical PMC result on the right side, a positive signal is obtained from a sample from Parkinson patient, whereas no signal is obtained from a sample from a healthy person. Finally, I will conclude by saying that the cerebrospinal fluid test for Parkinson's disease is being offered by Amprion now. However, blood test is still under development. I hope years to come, it may be available. I would like to thank finally my lab members, collaborators, and funding agency. Thank you very much for your attention.
Great. Thank you so much for a description of your research. I am now going to invite Dr. David Standaert to join us, who is an expert both in Parkinson's research and clinical care. And can you offer some commentary on the CSF test available and the potential for a blood test for Parkinson's to be available in the future?
Well, thank you, Rebecca. This is really exciting work. I think the idea that we're entering an era where we'll have blood tests or CSF tests or other kinds of tests for Parkinson's is really an exciting moment. One thing to appreciate is that this is happening based on our knowledge of what causes Parkinson's disease. This protein alpha-synuclein was discovered to be connected to Parkinson's about 23 years ago now. And we are finally beginning to understand the critical role it plays in Parkinson's, that these oligomers form, and these oligomers are bad actors. And they're part of the problem in Parkinson's disease. And the development of a test to test this or look for this in patients is a really important milestone in the field. Another thing this brings out is that Parkinson's is a whole body disease. We always think of Parkinson's as a disease of the brain, and it's true many of the symptoms that we deal with are related to problems in the brain. But here we're testing the blood and finding that there are abnormal proteins in the blood. And it really emphasizes that Parkinson's is a whole body condition and not just a brain disease. In terms of why would you want to test for Parkinson's? Can't we already tell who has Parkinson's and who doesn't? Well, the best test right now is the opinion of movement disorder experts, people like me who've spent a lot of years training and seeing patients with Parkinson's. And we're pretty good when people have had Parkinson's for a number of years and the symptoms are established. But we're not so great early on. And I'm sure many patients have had the experience of having trouble getting a diagnosis when their symptoms were early and mild. Parkinson's can be confused with other similar conditions, particularly early on. And a real need is to think about how early can we go? Can we actually detect these changes going on in someone before they have any symptoms of Parkinson's, before they have tremor, before they have any slowness? Can you find these symptoms developing really early in the condition? This would be a tremendous advance, because then we could talk not just about treating Parkinson's, but we could actually move to ideas around preventing Parkinson's, preventing the appearance of the physical symptoms if we could detect it really early. We've talked a lot about tests in the blood. As I said, the spinal fluid test is actually commercially available, and some sites are using this. It is available. Not all insurance companies will reimburse it at this point, so that's a challenge, but it is a test that can be ordered. You can talk to your physician about it. Now, I don't think that test is needed in most people who have well-established Parkinson's. It's far more useful, as I say, in the very early stages where there's questions about, is this Parkinson's, is it not, is it something else? That's really the role of this kind of test. Blood test, hopefully that's easier to get than spinal fluid, and hopefully we can see that reach clinical use soon. There are also other sources to be testing. There's a lot of work going on skin biopsy, using small pieces of skin and testing that as a source for looking for these oligomers. So I hope that this will allow more accurate diagnosis of Parkinson's. I hope that, right now, movement disorder experts are in short supply. I hope this enables testing to reach people who maybe don't have access to a movement disorder specialist. And I hope it pushes the timeline back and lets us detect this disease really early, in the hope that we can take steps to actually prevent appearance of the symptoms. So it's a really exciting moment in the field, and this is great work that's moving us forward. I was really excited to hear about it today.
Fantastic. Thank you so much, Dr. Shahnawaz and Standaert, for your presentation of your research and commentary. We're now going to ask Dr. Kevin McFarthing to join us. Dr. McFarthing, you are trained as a biochemist, had a career as a biochemist, and now have Parkinson's yourself. Now, you maintain a database called the Hope List, in which you have organized all the current research being done at all stages of the research pipeline. It is a vast undertaking. It's an extremely helpful document to understand the field of Parkinson's research. Having heard these two presentations and the commentary, can you share with us your perspective on the role of Parkinson's research in the lives of people with PD like yourself?
Thanks, Rebecca. As you rightly say, I maintain the Parkinson's Hope List because when I was diagnosed nearly 10 years ago, as a scientist and as someone who's what you might call an impatient patient, I wanted to find out what was happening in the areas of new therapy development. And today, that list extends to 370 projects. And I think that's the first thing that research brings us. It's hope. Hope for better symptomatic treatments for today, and hope for therapies that will slow or prevent the progress of the disease, stop it getting any worse. By the way, if you want to have access to the Parkinson's Hope List, just Google it and you should be able to find it. It's open source, free to use by anybody. Research also brings knowledge. Without an understanding of the basic biology, our progress is potentially built on foundations of sand. And research increases the chance of finding new therapies, improving old ones, and new ways to relieve symptoms and new ways of managing the day-to-day. Research can also inform the discovery and the choice of potential new therapies. In terms of methodology as well, clinical research can provide new ways to conduct preclinical and clinical studies. Because as we probably all know, clinical trials take a long time to complete. They take that time necessarily because safety is very important. Scientific integrity is very important as well. As we've seen from Dr. Shahnawaz's work, research can produce potential biomarkers that enable us to monitor the progression and to conduct more efficient clinical trials. We only know how we feel. We know how we feel can change from day to day, even from hour to hour. Scientists and clinicians can observe what we're doing and come to their own conclusions, but they need to try and make their subjective observations into objective science. And in order to develop therapies that can improve the symptoms and change the progression, we need biomarkers. We need very good biomarkers. If you have diabetes, you can measure hemoglobin A1C. If you have prostate cancer, you can measure prostate-specific antigen. We need our own hemoglobin A1C. So, even if a cure or cures, which is I think more likely to be the case, aren't available today, research brings the hope that they will be available for the next generation of people with Parkinson's. And for people with a family history of Parkinson's, that's particularly important. Finally, what I'd like to do on behalf of people with Parkinson's is to say thank you to the researchers who are dedicating their working lives and a lot more to trying to improve our lives today and trying to improve the prospects for the future. And with that, I'd like to hand back to Dr. Gilbert.
Thank you so much for Dr. McFarthing's comments and for everyone's comments. We are now ready for our Q&A. We've had many questions that have been submitted to us before the presentation, but we are eager to hear your questions currently. So people have already been putting their questions in the live chat, and we encourage you to continue to do so as we continue our discussion. And I will direct the questions as appropriate to our various panelists. So thanks for those incoming questions. Our first question is from Carolyn. And she asks, and I'm going to give this question to Dr. Standaert, what research is ongoing involving alpha-synuclein depletion and its impact on neurons and Parkinson's disease?
Well, there's a lot of interest in this idea of if the problem is too much alpha-synuclein, could we reduce the amount of alpha-synuclein? But it's not just a quantity of alpha-synuclein. As was discussed earlier, it's the abnormal forms. It's these oligomers and abnormal forms of synuclein. Synuclein is found normally in all of our bodies and brains. So we don't want to just reduce the total amount of synuclein. You want to try and reduce the abnormal synuclein. There are a number of studies. There are companies developing, for example, antibodies that could be infused once a month to remove abnormal alpha-synucleins. And these are pretty late in the clinical trial game at this point. So we're going to learn more about those soon. Other approaches to reducing the expression of synuclein, and then approaches to enhancing the body's normal mechanisms by which it clears alpha-synuclein. So I think all of these are potentially viable. They've all worked in animal models. And as I said, several, particularly the antibody approaches, are well into phase 2 and phase 3 clinical trials. So we're going to learn a lot more about these soon. I think that's an exciting area. And if you combine that with a blood test to identify somebody with abnormal synuclein early and then remove it, that to me is a potential prevention of Parkinson's. It's really exciting.
Fantastic. Thank you so much for that answer. Our next question is to Dr. McFarthing. Of all the trials that you have collected, can you enlighten us as what you think is one of the most promising ones that you're very excited about?
Well, one that I find very exciting is Anavex 273. Results of a phase 2 trial of this drug were announced towards the end of last year. It's what's called a sigma-1 receptor activator, which helps proteins to fold normally inside the cell. In their phase 2 study, they found significant improvement in cognitive measures. They also found a 14-point difference between placebo and Anavex 273 on what's called the UPDRS, which is the favorite way that clinicians have of measuring our Parkinson's symptoms. This is after 14 weeks of study. So this is excitement. On the other side, coming with a lot of cautionary notes, because it's a phase 2 study. We need bigger numbers in phase 3. The results haven't been published in the peer-reviewed journal yet, but the top line looks very exciting indeed.
Fantastic. That is always exciting to hear about other possibilities out there. We have another question from, let's see, who's that from? Can't find the name, but the question is, and this is for Dr. Shahnawaz, is the blood test for Parkinson's that you were talking about now engaged in clinical trials, or is this still primary research?
Thank you very much, Rebecca. Thank you very much for asking this question, because currently we are developing. As you know, that we have developed the CSF-based test, but right now we are working on developing a blood test. The main problem here is the complexity of the blood matrix and the extremely low quantity of alpha-synuclein oligomers in the blood. So we are addressing these problems, and we are working on different approaches to concentrate alpha-synuclein oligomers from the blood and to remove other contaminating protein from the blood. We believe that using PMCA, we would be able to detect tiny amounts of oligomers in blood, even other biological fluid like the saliva and urine we are working on. But this is currently in research. Right now, we are not going for clinical trial right now.
Okay, so we need a little longer before we can use actual people with PD to test whether they have Parkinson's through their blood, but it's coming, so that's very exciting. Question for Dr. Bentley. So focused ultrasound is another procedure that is currently available for Parkinson's disease motor symptoms. Is there any thought of using focused ultrasound for cognition in the future?
I would say maybe in the far future, but currently we know very little about cognitive networks. And focused ultrasound creates an irreversible lesion. So we would have to know where to lesion exactly and how that particular area works with the rest of the network, such as Dr. Weintraub was describing. I like the neuromodulatory aspects because you can turn stimulation up or down. You can change it not just in frequency, but as he alluded to, other parameters as well. So I think at this point, there's still a lot unknown to be able to use that sort of lesioning, but potentially in the future, as we learn more and have a better understanding, it could be possible.
Fantastic, thank you. Question for Dr. Weintraub. So we discussed cognition and how other brain regions or other frequencies of stimulation may be investigated for cognition in Parkinson's disease. Can you envision other symptoms of Parkinson's that deep brain stimulation may be able to help if we choose the right frequencies or brain targets?
Maybe a brief two-part answer to that. I would say within Parkinson's disease, there's similar research to what Dr. Bentley was describing, but that focus on psychiatric symptoms, let's say, and have demonstrated that the stimulation parameters, whether it's location or frequency, post-surgery may have differential impacts on depression and anxiety, for instance. So I think over the years, my impression is that the people that are doing the DBS programming, let's say the movement disorders neurologists, will have more tools available to them to make adjustments that may have beneficial impact on mood or cognition post-surgery. Beyond that, I'd say that DBS outside of Parkinson's disease is used in a range of psychiatric conditions, either experimentally or even clinically. So disorders such as obsessive compulsive disorder, major depressive disorder in the general population are sometimes treated with deep brain stimulation in areas different than are used for Parkinson's disease. There's also an interest in deep brain stimulation for the treatment of substance use or addictive disorders. So I'd say within psychiatry, DBS continues to be explored as a treatment. And since most of the psychiatric disorders that occur outside of Parkinson's disease can also occur within Parkinson's disease to varying extents, it may have applicability to Parkinson's disease one day as well.
Excellent. Okay, so there's a lot going on in deep brain stimulation and Parkinson's, a lot more to come. Very, very exciting. We have a question from Karen, which I'm going to ask Dr. Standaert to answer. This is a general question about research. So Karen says that her sister passed away from a long-term impacts of a young onset Parkinson's. Is it helpful to researchers to have Parkinson's listed on the death certificate? Is that something researchers have access to and can use for their work?
Well, I always think accuracy in death certificates is very important. We actually don't have really good data on exactly how many cases of Parkinson's disease there are in the United States or exactly how many people die from this every year. We have some rough ideas, and a few states have implemented registries, but for the most part, the data is a bit spotty. We do know Parkinson's is becoming more common just because our population is aging. But we're interested in whether there are other factors. Are there environmental factors? Are there other factors that may be changing the rate? And without good data, we can't really tell. And the death certificate is one important data point. That data is available for researchers. So I encourage people to try and promote accuracy in death certificates. I think it's a key tool for us as we go forward and try and understand Parkinson's and what may be causing changes in numbers in the population.
Thank you for that. We have another question for Dr. McFarthing. As somebody who is deeply involved in what's going on overall in the whole entire Parkinson's disease research world, are there elements of the research pipeline that you find most frustrating?
Good question. Yes. The general theme in clinical trials is the time it takes. I mean, it's reasonable to understand the reasons why it takes a long time, but the fact that it takes a long time, as I mentioned before, I'm an impatient patient. I'm certainly not the only one. There are lots of reasons for that. One of them could be that the pace of recruitment. It takes a long time to recruit people to clinical trials. It can take a long time to actually get them through the treatment, even if the treatment is a relatively short duration. Of course, one of the problems with looking at the progression of a disease like Parkinson's is one characteristic, which actually is good news. It is relatively slowly progressing. But if you're trying to measure the difference between a therapy and a placebo over a long period of time, to get a reasonable gap in the lines, if you're going like that with the treatment, if you're going like that with the placebo, it takes time to build a gap between the two that's statistically significant. So that's a frustration that applies to studies for most neurodegenerative diseases. And it's also frustrating that despite massive investment in time and money in disease-modifying therapies, we still don't have any. And probably the only thing that's been shown to influence the rate of progression is exercise. but there are many potential drugs in the pipeline that are looking to modify the course of the disease.
Very good. So lots of frustration, but lots of hope as well. So hopefully the trajectory is forward, and a neuroprotective agent is on its way. I have a great question for Dr. Bentley, which is from Fred. In deep brain stimulation, multiple leads be placed in different parts of the brain. So he's envisioning having deep brain stimulation to two brain regions, one to help motor, one to help cognition. Is this something potentially feasible?
That is a great question. And I would say the short answer is yes, that deep brain stimulator electrodes can be placed in multiple areas. Sometimes we do this currently in Parkinson's disease for severe refractory tremor, where there are two targets that we can choose. It's also used sometimes in other diseases to modulate more of the psychiatric indications that we've touched on. For this particular cognition plus the motor symptoms, it is an option that we can think about. Also, there are ways to cycle the stimulation. So as mentioned before, we definitely don't want to lose the motor benefit that patients are getting. So we think about things like administering sort of theta burst stimulation during sleep, for instance, when the motor symptoms are not as severe. But yes, there's all sorts of creative ways I think that this can be used for more personalized therapy.
Great, fantastic. Question for Dr. Shahnawaz. When this blood test becomes available for Parkinson's, what quantity of blood would you envision would be needed to perform this test?
Thank you very much again for good questions. We cannot be sure when the blood test will be available. We are working on that. The problem is, as I said earlier, that the quantity of oligomers is very low as compared to cerebrospinal fluid. There you have a lot of quantity of alpha-synuclein oligomers, and it can be detected very easily. The problem is here is not only the quantity, but also the complexity of blood matrix contaminating proteins. So we are currently working on to increase the sensitivity of the PMCA assay so that they can detect the small or tiny amounts of oligomers. So currently we are working on at least 500 microliter of blood test for blood plasma, and to get more, that will be better, at least 1 ml or 2 ml. That will be good to start with. So we are working on that. Main thing is we are trying to increase the sensitivity of the PMCA. If we have a very sensitive method, then you can even detect the single particle of alpha-synuclein oligomers. So we are trying to reach that. So we have done previously with the prion protein to detect a single particle. So we are trying to reach that level so that we can detect even the single particle of alpha-synuclein oligomers in the blood. That will be very useful. In that case, we don't need too much. 500 microliter or 200 microliter will be enough to start.
Fantastic. That sounds like a reasonable amount for a blood test. So we'll see what the future holds. We are at the end of our session, and just a question came in just now. Actually, this is a great question for to wrap us up for Dr. Weintraub. There's a question from Lorene, and it is, why is memory testing so important pre-DBS surgery?
I would say maybe a couple of reasons. One is we do recognize that some patients, fortunately it's a small minority of patients, can experience more meaningful cognitive decline post deep brain stimulation surgery. So we want to try to identify those people in advance, so to recommend that they not get surgery. And it seems that the people that have pre-existing cognitive impairment, so cognitive impairment prior to surgery, is a group that's at increased risk of decline afterwards. So that's why you need to do testing, and it needs to be detailed testing ideally, not just superficial testing, which may miss meaningful cognitive changes. So that's why we typically recommend that somebody meet with a neuropsychologist and go through 1 to 2 hours of detailed cognitive testing to make sure that it's safe to have them go through the neurosurgical procedure and the stimulation afterwards.
Fantastic. Well, we have reached the end of our question and answer session. I want to thank our panelists for all their wonderful information. I want to thank Dr. Shahnawaz, Dr. Standaert, Dr. Bentley, Dr. Weintraub, and Dr. McFarthing. I also want to thank everyone for participating in today's discussion and for your questions. So if you know someone who missed today's program, if you joined late, and if you'd simply like to watch this again, this recording will be available later today on our YouTube channel. And don't forget to subscribe to APDA's YouTube channel while you're at it to watch new videos and live broadcasts. At this time, I want to point your attention to the fact that the projects that we described today were both funded by APDA. And if you want to learn more about our research, please visit us at apdaparkinson.org/research. You can learn more about many of the other projects that we sponsor and fund. For additional information on resources, again, you can find everything on our website, apdaparkinson.org. The next episode of Dr. Gilbert Hosts will be on March 31st at 3 p.m. Eastern Standard Time. I'll be hosting movement disorder specialist Dr. Linda Nabobi, and we'll discuss at that point, I was diagnosed with Parkinson's disease, now what? And she'll answer questions about early signs of Parkinson's, how Parkinson's is diagnosed, how to navigate a new diagnosis, and more. So you can register now on our virtual calendar, apdaparkinson.org/events, and scroll to March 31st and register now. If you enjoyed today's program, we hope you'll consider supporting APDA with a donation, because with your help, we can deliver more programs and services like this one. I want to thank everyone again for joining us today. We hope to see you again on another APDA program. Have a wonderful afternoon.
Hi, I'm Leslie Chambers, the President and CEO of the American Parkinson Disease Association. Each month, across the country, APDA is providing support groups, exercise classes, and educational programs like this one to support the Parkinson's disease community. You can find all of our upcoming virtual events on our website at apdaparkinson.org/events. If you enjoyed today's broadcast, I hope you will consider making a donation to help keep programs like this possible. Your gift can help APDA support people living with PD through local programs, reliable resources, and groundbreaking research designed to find treatments and ultimately the cure for Parkinson's disease. Please donate today at apdaparkinson.org/donate. And thank you so much for your support.